we welcome the delegates to this particular session, and warm welcome to all the scientific experts of JTech Company and Cow Corporation from Japan. Welcome to all of you. The first session will be of JTech Company Limited, Japan. And I request, Dr. Nazir.
Yeah,
you can start now, please.
Thank you so much, Dr. Patil. So, good afternoon, everybody, and welcome to this very interesting session. So today I have been given this responsibility to begin with the proceedings of the day. We welcome all the delegates, and for this first session of the day, which happens to be session number 7 of the conference, we have representatives of JTech Japan, and the contents of this session are interesting in multiple ways, because many of you who were there probably during the satellite meeting also, when we discuss alternatives, we also know that there are certain challenges of getting alternative to animal models out there. One major challenge being the regulatory acceptance. Though we can use alternative model systems for generating proof of concept data, and at the basic level, when we study mechanistic understanding of multiple disease conditions and related aspects, we know that there's a wide acceptance because there's a large body of data that's out there. But when it comes to regulatory acceptance, particularly when we talk about drugs and diseases, and when we know that for filing an IND application, the application for an investigative new drug, there are certain regulatory guidelines which are very stringent, and not all of the new models and new methods have received that wide attention that they deserve and do not have regulatory acceptance as of now. Considering the contributions of JTech, I believe they bring something extremely interesting to the platform, because they talk about some models which have already received the OECD acceptance. So we'll know more from them, but that's extremely interesting. So today, Mr. Yoshihisa Yamayuchi and Dr. Hiromichi Mitake from JTech, Japan, will talk to us about human reconstructed epidermis, the lab site epi model, for alternatives based on regenerative medicine. And their company has been established in 1999, and their aim is to industrialize the field of regenerative medicine. And when I talked about the OECD recognition, they say that their reconstructed normal human epidermal model, namely lab site epi model, is listed in OECD TG439 under the in vitro skin irritation test systems, and TG431, which is for in vitro skin corrosion test, also has the OECD recognition. So we'll be looking forward to what they have to discuss about, because it's an extremely interesting field, and having OECD guidelines actually makes me also particularly interested about knowing more regarding this model system. So over to Mr. Yoshihisa Yamayuchi and Dr. Hiromichi Mitake for their presentation. Please go ahead.
Can you see our presentation?
Yes, we can see that.
Yes, we can, and you are audible, and everything looks perfect. So please go ahead.
Thank you. So hello, I am Yoshihisa Yamauchi from Japan Tissue Engineering, JTech. First of all, we would like to thank Dr. Akibasha and the board members of SAAEI for the opportunity to give us this presentation today. We also appreciate Dr. Kojima at JSAAE, who introduced SAAEI to us. Today's presentation is aimed to introduce our product, human reconstructed epidermis, lab site Epimodu for the alternatives based on regenerative medicine. So, do you know who we are? Yes, we are JTech, the pioneer of regenerative medicine industry in Japan. We have developed the regenerative medicine for more than 2 decades, and our product got approved at first by the regulatory authority in Japan. Now, we are manufacturing 4 approved products. It is the largest number in Japan. Our company's vision is creating a future for regenerative medicine. We strive in all kinds of fields to create a reality in which everyone can enjoy the benefits of regenerative medicine close at hand, anytime and anywhere, to maintain their health. This is outline of today's presentation. First, let me explain the company overview of JTech. This is the company overview. JTech was established in 1999. Since then, we have been researching, developing, manufacturing, and selling the approved regenerative medicine and the cultured human tissue models for research use. Our capitalization is about 5 billion yen. Our president is Kenichiro Hata. He is also the chairperson of the largest industry organization of regenerative medicine in Japan. We are widely recognized as the pioneer of regenerative medicine in Japan by the pharmaceutical companies and the regulatory authorities. JTech headquarters is located in the neighbor Nagoya, the central area in Japan. Our parent company is Teijin, the global textile and chemical company who is also putting effort in the healthcare business today. Teijin has 2 business bases in India, at Gurgaon and Pune. JTech and Teijin are now collaborating to spread our products in India. We have 3 business segments. The main is regenerative medicine business. Our products got the approval from Ministry of Health, Labor, and Welfare in Japan. We are manufacturing and selling under the strict legal restraints, the Pharmaceutical and Medical Devices Act. We also run the contracted development and manufacturing business of regenerative medicine from the partner company and academia. These sales are almost all domestic because our regenerative medicine products are approved only in Japan. And we apply the technology and know-how of regenerative medicine to the products for research use. This is lab-site series, the human 3D cultured epithelial cells and corneal epithelial cells. We promote the alternative to animal experiments through the providing these products in Japan. We sell these products not only in Japan, but also export to nearby Asian countries. We intend to expand the sales of lab-site series overseas with the support of Teijin. Our sales result of fiscal year 2020 is $21 billion. Then, let me introduce our regenerative medicine products. Today, 14 regenerative medicine products are approved in Japan, and 4 of them are ours. This is JACE, the autologous cultured epidermis. JACE is the first approved regenerative medicine in Japan. Thus, we are recognized as the pioneer of regenerative medicine in Japan. JACE is used to treat severe burns, congenital giant pigmented nevus, and epidermolysis bullosa. We collect a small piece of patient's own skin culture cells from that skin to make the seeds of cells like this picture. And they are transplanted to the patient. And these are other regenerative medicine products, which are all derived from autologous tissue. JAG is the autologous cultured cartilage, which is the second approved regenerative medicine in Japan. JAG is used to treat traumatic cartilage defect at knee joints. And NEPIC is the autologous cultured corneal epithelium. NEPIC is used to treat limbal stem cell deficiency for the patients whose stem cells around the cornea are heavily damaged by the chemical leakage or inflammation and lack their corneal epithelium. Ocular is the autologous cultured oral mucosal epithelium, the latest our also approved product. Ocular is also used to treat limbal stem cell deficiency, especially for the patients who lack limbal stem cell theory. Oral mucosal epithelium can function as the substitute of corneal epithelium. We are dedicating ourselves to developing the regenerative medicine products more than 2 decades and provide unique treatments to the patients who could not be saved in the past. And this is our facility. Approved regenerative medicine products are manufactured at the production area, where the cleanness of the environment are highly controlled. Production facility is inspected by the regulatory authority and proved to match the regulatory standard of GCTP, good gene, cellular, and tissue-based products manufacturing practice. Thank you.
Okay, this slide presents the switch. I am Hiromichi Mtt, working at JTech, the sales representative. Next topic is introduction of lab site. Okay, we have developed lab site by applying technique and know-how of regenerative medicine products. Lab site has 2 lineups, skin model and cornea model. Skin model named as Epimodu is listed in OECD 439, which is skin irritation test, and OECD TG 431, which is skin corrosion test. Cornea model is also listed in OECD 492, the OECD guidelines for testing of chemicals are correction of the most relevant internationally agreed testing models. So lab site is widely used in Japan for alternatives to the animal experimentation. There are 2 types of Epimodu, 12 well types and 24 well types. The shelf life of Epimodu is 5 days at room temperature. At present, we ship our model almost every Monday. So Epimodu can be stored until Friday of the shift. The OECD listed model is Epimodu 24. This slide shows the gross appearance of Epimodu 24. The tissue is reconstructed in the cell culture insert like this. Cell culture inserts are placed in the culture plate. The human epidermis are reconstructed in the insert. Although they, before cell seeding, bottom of cell culture insert transparent, the bottom of cell culture insert turned white when tissues are reconstructed. This is a model structure of Epimodu. Please see this figure. We have 2 types of Epimodu, 12 types and 20 well types, as I mentioned. The diameter of Epimodu 12 is 10.5 millimeter, and that of Epimodu 24 was 6.4 millimeter. The top of the cellular layer is exposed to the air, and meanwhile, media is filled in the culture plate. There, pour the membrane filter, and cells are ground with basal surface in the contact with media, resulting epidermis is reconstructed on the membrane filter. This is histology of Epimodu. The left side is historical image of normal human epidermis. Whitish part is dermis. Epimodu is a reconstructed epidermis. So this pink part is equivalent to the epimodu. The epidermis consists of 4 layers from top to basal, conifer layer, granular layer, spinous layer, basal layer. You can see the differentiated 4 layers in the epimodu, as well as sorry. So you can see the differentiated 4 layers in the epimodu. The structure of epimodu is similar to that of normal human. This is the histological changes during 4 weeks culture self-seeding with day is day 0. So this 1D means that the next day after self-seeding, the first cells are layered gradually like 4D. The conifer layers appeared around day 6. So you can see the dark pink part, which is conifer layer in the 7D picture. After formation of conifer layer, the thicker and thicker by culture, like this. There are differences between human epidermis and epimodu. Epimodu is reconstructed from only one kind of cells, keratinocyte. So there's no appendicular organs such as hair follicle and perspirant glands. And the turnover is not occurred in epimodu. So conifer layer is thicker and thicker by culture. The epidermal stem cells are also confirmed basal layer in the epimodu. However, the activity of the stem cell in epimodu is not maintained by our culture technique. So viable layer is thinner and thinner as it cultivates like 28 days. After cultivated 13 days, epimodu is shaped. So the condition of epimodu is almost equivalent to the picture 4D. Next is quality control of Epimodu 24. We have 3 standards for the quality control. First, we check the cell viability of Epimodu every lot. This figure shows the result of 10 lots. We measure the cell viability by Mtt Assay, which is colorimetric method. The vertical axis is OD value, horizontal axis shows the lots. Tick bar graph is a mean of every lot of the OD value, or lots fall with the range of 1.4 to 1.6. The error bar is very small. Next, we evaluated barrier function of Epimodu by IC value, IC50 value. This is a data for the 10 lots of Epimodu. Vertical layer is cell viability, and horizontal axis is the concentration of SDS. The concentration SDS is 0.1 to 0.4. The IC value is here. The IC value for the all 10 lots are near the 0.25, and near 10 lots are near 0.25%. We also perform histological assessment every lot. Until now, all lots of Epimodu pass these 3 standards. we have never confirmed the non-conformance lot. Considering those, we think we can produce lot-to-lot in lot variation products. Okay, let me summarize that from my presentation here. Point one, lab-site epimodel is reconstructed the future of 3D tissue exhibiting reproduction of human epidermal tissues. And point two, Epimodel 24 is listed in OECD 439 and 431. So researchers can perform the skin irritation and corrosion test according to OECD TGs. Point three, we developed the lab-site epimodel by applying technique using the manufactured cells-based product for regenerative medicine, resulting that we can produce lot-to-lot in lot variation. In this slide, I would like to explain the protocol OECD 439 using Epimodel 24. First step is test chemical application. After received products, tissue are incubated for 15 to 13 hours. Then test chemicals are applied on the surface of tissues. After exposure of test chemical for 15 minutes, test chemicals are removed and the tissues washed by PBS flow. Lastly, tissues are post incubated for 42 hours. Second is Mtt Assay. After post incubation, assay medium are replaced with MTT medium and tissues are incubated for 3 hours. If tissues are living, tissue reduce the parmesan and the color of tissue turned purple, like this photo. Third step is quantification of the living cells. Parmesan is extracted by isopropanol. The absorbance of extract at 570 nanometer and 650 nanometer are measured. The cell viability of samples are calculated. Cutoff value is 50%. If cell viability of the sample are 50 and less than 50%, the samples are classified as irritant. On the other hand, the sample which viability are above 50% are classified as non-irritant. This is a concordance with in vivo classification. We researched the concordance in TG439 using our model. We use 54 test chemical substances classified in vivo in the past. In vivo, 70 substances are classified as irritants. Among them, 60 of 70 substances are classified as irritant in vitro. So sensitivity is 16 divided 70 equal 94.1%. Meanwhile, 37 substances are classified as non-irritant in vivo. Among them, 26 of 77 substances are predicted as non-irritant in vitro. So specificity is 26 divided 37 equal 70.3%. Finally, 16 substances and 26 substances are predicted correctly, resulting accuracy is 77.8%. Next is TG439 in vitro skin corrosion test using Epimodel 24. The procedure is similar to TG439. First step is test chemical application. The exposure time is 3 over 60 minutes. After application, test chemicals are removed by rinsing the tissue. And in TG439, post-incubation is not needed. The washed tissues are for the next step, Mtt assay. The cell viability are calculated by Mtt assay. This slide shows the concordance of TG431 using Epimodu with in vivo classification. Our model allows the subcategorization processes according to cell viability. The test chemicals are classified as non-corrosive 1B and C, 1A. The sensitivity of this method is 100%. The rate of accurate classification for corrosive is 95.6%. The rate of perfect agreement with in vivo classification is 78.9%. From this slide, next I would like to introduce the Epimodu 60. 60 means 60 culture model. Regular model is cultured for the 13 days as I mentioned. So culture period 60 model is 1 week shorter than regular model. That resulting that 60 model exhibits in a conifer layer compared to regular model, like that. Conifer layer plays a role for barrier function, so you can detect latency of substance more sensitivity using Epimodu 60 model. In Japan, most 60 model customers use this product for the developing the product for baby care and sensitive skin. This is, for example, using Epimodu 2460 model. Generally, cosmetic exhibit low irritancy. The safety of cosmetic is well investigated. So most cosmetics are classified as non-irritant in TG439. Please see this table. Our customers, researchers at Nihon Koruma performed the TG439. There are 10 cosmetic features evaluated by human patch test in the table. The 50 minutes exposure and 24 hours post-incubation and Mtt assay is the protocol of TG439. All of tested cosmetics classified as non-irritant. Even using 60 model, the judge of these cosmetics are not changed. The situation like this is not good for developing cosmetics. So they changed the protocol exposure time 15 minutes to 24 hours and remove the post-incubation. So some of the substances are classified as irritant using Epimodu in their method. While in the case of using 60 model, they demonstrated that 60 model can detect the substance more sensitivity. For example, lotion 1 is classified as irritant, modified protocol using Epimodu and 60 model. Lotion 2 is also classified as irritant in only 60 model. In human patch test, lotion 3 is higher score than lotion 2. So this method prediction don't completely match the score of the human patch test. But Nihon Koruma identified the irritant substance using this method when they could receive the complaint about the irritancy of their product on the market. Finally, they changed the formula of the product based on the result of this method and successfully placed the improved low irritancy product on the market. Although this method does not exhibit completely prediction for the human skin, but it is a useful method depending on the situation. This is novel test. Next topic. This is a novel test method for skin sensitization test using Epimodu. This method, named as Epsensor, is developed and reported by Kao Corporation, which is a famous Japanese company. The second report showed Epsensor exhibit good reproducibility within and between laboratory. This assay based on the induction multiple marker genes related to keratin site response, inflammatory cytoprotective. If sensor will be listed in OECD TGs, you can perform the three methods according to OECD TGs using Epimodu 24. Last topic is I would like to explain the situation of the lab site users in Japan and the Asian countries. In Japan, we have most of customers in the cosmetics and daily necessity industries. They use our product mainly evaluation of safety tests. Of course, some of cosmetic research evaluated the effectiveness of their products. Also, academic users and CRO users use our products. The academic researchers mainly perform basic research, and CROs basically perform the experiments according to OECD TGs. As I shown in this slide, Epimodu is widely used across the industry in Japan. This is domestic share Epimodu in Japan. In Japan, three models listed in OECD TG 439 are available. This A and B are the products from other company. Among them, the share of Epimodu is 58%, and we gained the top share in Japan. As this slide shown, Epimodu is the most popular reconstructed human epidermis in Japan. Although Epimodu is the most popular product in Japan, but there are not so many customers in the overseas. At present, Epimodu is used in some Asian countries around Japan, such as Korea and Taiwan and Thailand, Malaysia and Singapore. Next, we would like to spread our product to India. Our aim is promoting and propagating the alternative Asian country using our technology based on regenerative medicine. As we mentioned previously, our group company is in India, Teijin Frontier Private Ltd. They supporting us promote the alternative in India. So we hope Epimodu is used by many researchers in India. This is our company web page. Technical information of Epimodu is in web page. Our vision and strengths as a regenerative medicine products company are also described in this homepage. If you are interested in our products, please access to our website or contact us. That's all. Thank you for your kind attention. I would like to thank Dr. Akbasha and the board member of SAAI for giving us this opportunity again. Thank you.
Thank you, Dr. Yamauchi and Dr. Mitake. That was indeed very interesting presentation. So we have a few minutes for the next session. So I would like to open the discussion with a question that I have. Since you mentioned that this is successfully being used and you presented some interesting data also on the accuracy and sensitivity as well. My question is, I mean, has this test been used outside Japan also successfully for getting any of the products, taking the products forward? And did you get my question?
Sorry, once more again.
Dr. Mitake. So I want to know, I mean, since you say that this is already listed in OECD, right?
Yeah.
So obviously after OECD now, the products that are coming to market might be using it for getting regulatory approvals. Is that correct? Within Japan?
Yeah, yes.
Right. So outside Japan also, any of the products that have successfully used it and obtained regulatory approvals based on conducting these tests?
Sorry, it is difficult.
Okay.
So, yeah,
Dr. Amir, you can
Hiromichi.
write questions so he can understand better.
Yeah, Dr.
Chat box.
Hiromichi, let me translate it to you. Dr. Amir wants to know whether your test is being used outside of Japan by regulatory authorities for allowing the drug product or allowing the regulatory clearance.
Whatever it is.
Yeah, that's what he wants to know.
Sorry, I don't know whether our products are used in the overseas according to their own countries. So maybe customers overseas use our model mainly to perform the OECD TGs. Okay.
Okay. So within Japan, are you aware that, I mean, any of your customers who have obtained the test system from you and they applied for regulatory approval based on data generated with your system, and they were granted a regulatory approval? It could be a drug, it could be a cosmetic. So has that happened in Japan? I mean, you have data?
Yes. In Japan, customers use our product according to the regulatory. So some of the testing for cosmetics and medical device are approved in Japan using our model.
Okay. So my last question, that after conducting this in vitro test that you have developed, is it mandatory to have any further validation before approval is granted? Wherever this is recognized, is another validation mandatory, or just on the base of your test, where I see that the endpoint happens to be Mtt Assay, right? And cell viability. So once one provides the data out of these tests on the Mtt Assay and cell viability from this in vitro test, if I have a product, I conduct this test and provide data to the regulatory agency, would they ask me for a validation, or is it just a preliminary test, or is it a final one? Would validation be required or not using any other system? That's my question.
The final test, according OECD TGs according to cell biopsy is a final test.
Okay, it's final. Okay, got it. So no further validation required. Approval granted based on this. As far as the skin irritation test and others. Great.
Okay.
Thank you so much. Thank you. So the forum is open for discussion. If there are any other queries, we have a few more minutes. Please go ahead.
Hello.
Yes, Dr. Sridhar, go ahead.
Yeah, I just want to know the academic purpose. We can actually utilize this model system for proteomics and metabolomic studies, or only we can do toxicity evaluation with this model.
Okay. In Japan, some of our customers perform the omics using our model, but the data of their data is confidential, so I don't know it is good performance for their product, their aim. Sorry.
Okay, we can utilize for marker studies, like the same model system we can use, like specific marker, epidermal marker analysis.
Generally, some of our customers perform the deep sequencing and targeting their product, targeting their protein and performing performing their experiments according to targeted protein. Okay.
Okay, thank you.
Are there any other questions? Okay, it seems none. So then
I think.
Yeah, Dr. Akbar Shah, please go ahead.
Yeah. Just introduce me to the speakers. I am Akbar Shah, the General Secretary of the Society for Alternatives to Animal Experiments. So I'm getting introduced to you for the first time, and I'm happy that you are here, making the presentation and explaining the product of your company. I just want to have a clarification of the same question which Amir and someone else asked, but in a different perspective. Like, so your product is approved by OECD for 431 and 439, and even 491, and it is regulatorily approved for use in Japan for irritation and corrosion, testing product for irritation and corrosion. Now you have a franchise in India. Am I right?
Yeah.
Is it a franchise or it is an outlet for marketing outlet in India? A Teijin, is it? Is it Teijin?
Yes. So we have a subsidiary company.
Yes,
here.
subsidiary company. Okay. So they market your product in India. So my question is this product or are your products regulatorily approved in India? Like our drug regulatory authority or cosmetic regulatory authority, have they approved your product to be used in the toxicity testing of cosmetic products or drugs? Or is it enough? The OECD approval is already there, therefore any country can use it.
That's a good question.
Controller General of India, DCGI.
DCGI. In India, it is the DCGI.
Right. Okay, there is a OECD committee in India. Actually, it's a committee. There's a subcommittee in India for the OECD that has approved these GLP labs. Any GLP lab, for example, Advenos or Bionides or anything who do the toxicology testing are able to do it because it's under OECD umbrella. We also have an OECD umbrella approval.
India is associated.
It's an OECD country. It's an OECD country. So separate anything approved in the OECD, we can use any as a toxicologist, if I want to apply this model, I can apply this model. I don't.
Mangala is right.
Yeah, Mangala is right. Yeah, that's right.
Right,
yeah.
Right. Yeah. So, Dr. Akbar Shah, I can add on to that. So, considering, I mean, the Drugs and Cosmetics Act, which of the DCGI, with the current guidelines that we have would obviously not include because they keep updating those guidelines and they were updated last year also after the previous revision in 2005. So as of date, I see that, I mean, such tests which we have proposed might not directly be included within those guidelines DCGI. But as Dr. Wangla correctly suggested, that once it is there in OECD, so that was, in fact, that was my question to the representatives, that if they can collect such data from other countries as well, which might not have immediately included in the regulatory framework and guidelines of their country, but this being OECD and OECD has worldwide acceptance, so they can collect that data also. That would be rather value addition for the product towards proposing to other people also that this has been used in these countries proposed. Because now, for example, if I am to propose a product and file for regulatory approval here in India, and I conduct this test, even though it does not appear in the DCGI guidelines as of date, I then cite that this is as per OECD. And then when regulatory bodies review my data, if I am granted an approval, because that time what I have to do is convince the regulatory body that this is done as per OECD. And once they give an approval, that obviously would be a value addition for their product as well, that this is as per OECD, and within so and so countries also, the regulatory approvals have been granted. based on the data that was generated from the study.
So let's not confuse the DCGI and the OECD because
That's
they're
correct.
two different organizations. So DCGI is only for India approval.
Right.
OECD, if I am providing service to a foreign country company or where they want me to do the toxicology testing, this country under OECD umbrella, we are covered, so I can do this experiment under OECD guidelines and submit the data to the country who's requesting me the.
Absolutely. When
there's no DCGI is not involved in this process.
It's not in there, obviously.
Submitting for something in Indian approval, you do need DCGI approval.
True. So
that was the question precisely.
Study for foreign country, but when we have to do it within our country, ultimately the data with whatever guideline we follow, the application has to be submitted with DCGI, right? So for example, it's a drug molecule here. So within India now, the DCGI regulatory body would be reviewing. So if it does not appear in their guidelines as such, but when given a reference of OECD, then they will review the data, and when convinced, they can give an approval. So this is about when we have to take approval within India. So we're talking this in Indian context here, that if we say procure their model, do a study over here, and then have to seek an approval from Indian agencies. So in that scenario, but of course, if it's for a foreign country, so for example, we procure these cells from Japan, and now we have to give approval for a company which is based in China. So that data could be submitted to them, and they can submit it to their regulatory bodies as such. Is that right, Dr. Mangala?
Yeah, I understand, and Dr. Mangala has made it clear.
OECD for other countries, not for India. DCGI is for India. That's it.
Yes, that's it.
Hello, sir.
That distinction should be very clearly made.
Sir, related to this, I have a question. Like,
we
what
can
about
one last question, because it's 3:14. So one last question before we move on to lecture. Go ahead, please.
Hello, related to this, I have what about the Ayush product? That formulation we can test it with this one, and I can utilize this specific model system. Ayush formulations like Ayurveda and other products.
You need to get Ayush approval, not OECD.
OECD approval or even DCGI approval.
Right, DCGI.
Ayush approval.
Ayush approval or even FSI. FSI is also regulating some of the Ayurvedic as well as food and nutraceutical products. So you don't need DCGI for those kind of products. Any Ayurvedic products should get either FSI or Ayush approvals.
Can I get any advantage with this? Suppose I am using this skin product.
Right. I mean, as long as that's a study that you're sure you need to be covered under, because if you go to Ayush website or FSI website, they have guidelines and say that these are the products that we will approve as nutraceutical or food products or Ayurvedic products. These are excluded from it. You have to go to DCGI for those things. So you need to go and look at those websites very clearly.
Okay, thank you.
We will wrap up. It's time.
Okay.
Amir.
So with this, we would end this session. It's 3:15, so we just are like 5 minutes over the time. So I think we largely kept the time. I would like to thank the speakers, the representatives of JTech, Mr. Yoshihisa Yamayuchi and Dr. Hiromichi Mitake. Thank you so much for the interesting perspectives that you shared with us. And we would like to, if any of our attendees today would like to obtain further information, we'll get in touch with you. And we hope you will be able to provide information and move forward. Thank you so much. Thanks to all the attendees, and thanks to the organizers, Dr. Akbar Shah, Dr. Rais, for giving me this opportunity, for chairing this session. So looking forward to other interesting sessions as well. Now over to Dr. Wangla for conducting the next session. Dr. Wangla.